Good afternoon, everyone. Thank you so much for joining us today. My name is Esperanza Stanchoff with Maine Extension and Maine Sea Grant, and I'm also a steering committee member for both NECAN and Maine Ocean and Coastal Acidification, or MOCA, partnership. I'd like to welcome you to the second webinar in the NECAN Sea Grant webinar series. In 2016, the Northeast Sea Grant Consortium, in partnership with the NOAA Ocean Acidification Program, funded 4 research projects to address the impacts of ocean acidification on key resource species in the Northeast as an aid to assist coastal communities in adapting to current and future OA conditions in the region. The research and observational priorities defined in the publication of a NECAN article in Oceanography titled Ocean and Coastal Acidification off New England and Nova Scotia were used in the 2016 request for proposals. The 4 projects chosen and funded are in Connecticut, Maine, and New York, and each of these webinars in the Sea Grant series are focused on one of these projects, their successes, challenges, and results. The full webinar schedule can be found on the NECAN website, along with the researchers' bios, and each will be recorded and posted there as well. Throughout the presentation, if you have any questions, you can submit them through the Questions tab on your GoToWebinar control panel, and we will go through each of those during the Q&A session following the presentation. So today we'll be hearing from Dr. Richard Wahle and Maura Niemisto on the gene response of post-larval lobster to in-century temperature and PCO2. Rick Wahle is the director of the University of Maine Lobster Institute and research professor in the School of Marine Sciences. He's based at UMaine's Coastal Laboratory, the Darling Marine Center, where he teaches and conducts research. He received his PhD in zoology in 1990 at UMaine, and after a couple of postdocs, served as a senior research scientist at Bigelow Laboratory for Ocean Sciences for 15 years before taking his current faculty position in 2009. Rick works at the crossroads of marine ecology and fisheries science. He founded the now long-standing American Lobster Settlement Index, a U.S.-Canadian monitoring collaborative and database of lobster nursery grounds that has proven to be instrumental in predicting trends in the fishery. A NECAN member from its beginnings, Wahle was recently appointed to Maine's newly formed Governor's Climate Council. And Maura Niemisto, she received her master's degree in marine biology from the University of Maine in August 2019 after studying the response of larval lobsters to in-century warming and acidification. Now she works as research associate at Bigelow Laboratory for Ocean Sciences, studying zooplankton physiology and behavior under Dr. Fields. Previously, she has worked as an environmental educator and tall ship sailor and maintains an interest in learning about how the smallest animals make a large impact on our marine systems. And with that, I will hand this over to Rick and Maura for their joint presentation.
Thank you, Esperanza. I trust you can hear me.
Yes.
So I'll continue as long as you can hear me. Well, it's absolutely my pleasure to deliver this webinar as sort of a continuation of several seminars and webinars we've given on the American lobster in a changing climate. Maura and I are here today on behalf of our collaborators listed here, and I'll introduce them in more detail in a few minutes. But I'd like to take about 10 minutes to set the stage for Maura's presentation, which is really the feature presentation here, on her NOAA-supported thesis work on gene regulatory responses of post-larval lobsters to end-century warming and acidification, as indicated here in the title. You know, a lot of this started back in around 2015 with the Maine legislature's establishment of the Maine Ocean Commission, and their goal number 1 was to determine the impact of ocean acidification on commercially important species and their mechanisms. And really the takeaways from that,
Rick,
yes.
I'm sorry, we can't see your screen.
Oh.
So I think you accept.
Oh, show my screen. Okay. Huh. I'm sorry.
Oh, okay.
Let me go back to this again.
Okay.
Can you
Now
see
we
my
can see it. Yes, thank you.
All right. Sorry, we had it all set up so nicely. In any case, you now see the you now see the title screen there with the title, Maura's and my name, and the collaborators listed there. I won't go through the whole initial introduction there, but I did want to establish the fact that Maine's Ocean Commission report in 2015 was really at the foundation of this, along with federal mandates for OA work. And the real takeaways from this was that our fisheries are dominated by marine calcifiers, that the Northwest Atlantic and the Gulf of Maine are warming and acidifying faster than many other parts of the world, and we really know little about their long-term effects. So let's just look at lobsters by the numbers, and this is all part of sort of setting the stage here. The American lobster is at this very moment the #1 fishery in the U.S. and Canada by landed value. About 90% of that value of the U.S. share comes from the Gulf of Maine, and about 80% of the U.S. share comes from Maine alone. It's also Maine's #1 export commodity, and about 3/4 of Maine's fishery value comes from lobsters alone. And this really reveals what I see as a perilous dependency on not just lobster, but on marine calcifiers. Lobsters, again, takes up more than 3/4 of the value, but then there are other contributors such as clams, scallops, oysters, even urchins that are also marine calcifiers and that add up to more than 80% of Maine's fishery value, along with lobster. So a perilous dependency. But how did we get here? Well, the NOAA trawl survey data illustrate this really well. These are the trawl survey data from federal waters going back to 1967, and you can see how coastal Gulf of Maine and Maine in particular and Southwest Nova Scotia just become a hotbed for lobsters by the 2000s and up to into this decade. And so what that meant for Maine is that we went from about 10,000 metric tons a year and increased that some sixfold, so that by 2016 we saw our peak value at about $500 million, and that doesn't even mention the added dollars and added value that's gained every time lobsters change hands down the supply chain and off to their ultimate market, as well as the other industries that depend on it, such as everything from tourism to trap making. But what are the ecological drivers of this change? The ecological environmental drivers. Well, we certainly know that the depletion of predatory ground fish has played a role, and that's shown really well in the chart of ground fish and lobster landings you see here from Maine. where while lobsters in the 50s, 60s, and 70s certainly were an important fishery, really the fishery was dominated by groundfish back in the day, and that means not just codfish, but various species of flatfish, monkfish, hake, and so forth. And by the 1990s, that began to change dramatically, essentially releasing lobsters from predation as we saw this dramatic, incredible shrinking fish, as it were, as we depleted the big predators out of the system. So certainly top-down effects have played a role in making things favorable for lobsters in the Gulf of Maine. But we also know that warming has played an important role, and unless you've been living in a cave for the past 5 or 6 years, you're now aware that the Gulf of Maine has been warming faster than most other parts of the Earth, or parts of the ocean, as we know from Andy Pershing's work, and that rate of warming has only been accelerating in the most recent years. It's also really important to know where lobsters fall in terms of their sweet spot or Goldilocks zone along the coast. And it's important on this really steep thermal gradient that we have from the Bay of Fundy down to southern New England, show where their thermal thresholds fall. And it's important to talk about two thermal thresholds, the high one being around 20 degrees C. Above those temperatures, we see physiological processes failing, and also the onset of shell disease in higher rates. At 12 degrees, I'll just back up here for a second. At 12 degrees, any temperatures below that tend to inhibit larval development, and that can mean the difference between populating and not populating nursery habitat along the coast. So as time has progressed, these isotherms have shifted northward, and so the same warming process that is adverse in southern New England and creating problems with shell disease has been really favorable in northern New England, and especially that eastern Maine coastal current area that has historically been on the cold side of the comfort zone. So those are thermal effects and top-down effects, but what about ocean acidification effects? Well, over the past decade or so, we've seen a number of helpful reviews of the exponentially growing numbers of papers on the topic, everything from reviews on OA effects on crustaceans to a focus on New England and Atlantic Canada, and even broader perspectives. Their collective takeaway message, if I can be so bold to make one, are these bullet points here. We see that ocean acidification effects are species and life stage specific, that crustaceans as a group tend to be relatively resistant to ocean acidification effects, but to date, mostly acute toxicity studies have been done. That's been sort of the low-hanging fruit, if you will, of studies that have first taken off. But the tougher nuts to crack have been looking at interactions of ocean acidification with other stressors, and we're just starting to look at the effects of OA on gene expression, their longer-term effects and intergenerational effects, most of which are still unknown. So that brings us to NOAA-funded project that we've been involved in since 2017 on the response of larval American lobster to ocean warming acidification across New England's steep thermal gradient. And our research team includes, in addition to me, who brings sort of the lobster expertise, if you will, David Fields from Bigelow Laboratory, expert in zooplankton ecology, and has been hosting much of the thermal OA treatment experiments over at Bigelow Lab. We also have Spencer Greenwood from the University of Prince Edward Island, who's really the genomics expert here, has done a lot of work with gene expression work with lobsters and other crustaceans. And of course, we also have Jess Wahle, who came on board just toward the end of her, on this project, just toward the end of her master's. Fraser Clark worked with Spencer Greenwood, now is at Dalhousie, so another geneticist. And of course, Maura here essentially took the baton from Jess after Jess published her work in 2017. And so we'll hear about Jess's work today. But the two big objectives of the overall project are to evaluate the joint effects of end-century warming and acidification on larval survival, development, respiration, behavior, and gene expression. It's a mouthful. We'll hear about a piece of that today. And to determine differences in the response among regional subpopulations among these three oceanographically contrasting areas. So today we're going to hear about from Maura about primarily her work on the gene regulation aspects and focusing on the results coming out of southern New England. And bear in mind that this is a work in progress, so we still have the other areas to evaluate. So Maura, it's off to you.
Great, thank you, Rick. Let me just make sure we're good and can see my presentation.
Yes.
Great, perfect. So I'm Maura Niemisto, and I just finished up my master's degree looking at a bunch of different responses in larval lobster to joint PCO2 and temperature stresses. But I'm going to be focusing just on gene regulatory response today. So we're talking about, let's see, changing ocean. Atmospheric concentrations of CO2 have increased 40% since the start of the Industrial Revolution, and this rate has only increased in the 20th and into the 21st century. And this is leading to important changes in our marine ecosystems. The IPCC just came out with a new report outlining the projected changes in oceans and the cryosphere, and this kind of paints a dire outlook, particularly for northern latitudes. This is a graph from that report showing the projected change in average global sea surface temperature relative to an average of temperatures between 1986 and 2005, which is the yellow line there. There are two separate emission scenarios highlighted here, red being the RCP 8.5, which represents a continued growth of greenhouse gas emissions, and RCP 2.6 that indicates a reduction in the amount of emissions being released now. So what's important is that in both scenarios, there's an increase in temperature between 1 and 2.5 degrees Celsius in average global sea surface temperature. And on a related note, as a portion of that CO2 is absorbed into the ocean, we expect the oceanic pH to drop as well. Again, this is the same report, and under these two emission scenarios, we should be seeing a drop of 0.1 to 0.3 pH units in average oceanic pH. And these are global averages across the ocean, but we are expecting to see more extreme drops in higher latitudes, particularly in the northern hemisphere and within our NECAN region. In fact, the Northwest Atlantic and Gulf of Maine is especially impacted by these changes. Rick already spoke to this figure here from Andy Pershing's paper that indicates that Gulf of Maine is warming faster than 99% of the world's oceans. Moderate emission scenarios estimate about a 3-degree increase in the Gulf of Maine's sea surface temperature by the end of the century. And as if that wasn't troubling enough, the Gulf is also vulnerable to acidification. It has a very low buffering capacity from many freshwater inputs and influence from the Labrador Current, which is very fresh. This is a figure from that paper that Esperanza cited earlier, showing the monthly mean minimum pH within the Gulf of Maine. It already sits low, and there are particularly low spots near the coastline and river outputs where a lot of these fisheries are. And as Rick already pointed out, this is extremely troubling for marine calcifiers. Now, many of you know the story of acidification already, but just in case there's somebody here who doesn't, I'll blow through it quickly. The absorption of CO2 into the ocean alters the chemical properties of seawater. Dissolved CO2 interacts with water to create carbonic acid, which in turn breaks down to create free hydrogen ions and bicarbonate ions. The presence of these hydrogen ions lowers the pH, but also they can bind with free carbonate ions in order to form even more bicarbonate. And the result is less biologically available carbonate, which calcifiers need in order to build their hard structures. And this can even dissolve some hard structures of some of our more vulnerable calcifiers. So in the Gulf of Maine, where we're heavily reliant on calcifiers, this is a problem. And to complicate this issue even further, how different creatures are responding to acidification is complicated and varies by species. So this is a figure from Croker et al. Showing metadata analysis on different taxa. In terms of survival, calcification, and growth, which are pretty standard parameters that are measured in a lot of these studies that look into ocean acidification. We have different taxa on our x-axis, so calcified algae, coral, echinoderms, crustacean. And on our y-axis, we have basically net change, positive or negative, within these different parameters. So you can see there's variation in how these taxa respond to ocean acidification. Coral, for example, tend to be very affected negatively, whereas crustaceans tend to be relatively resistant in these measured parameters of survival, calcification, and growth. But a lowered pH also affects organisms' physiology in other ways as well. As it can cause metabolic depression, reduction of energy stores, and a redirection of energy from biological processes in order to maintain functioning of these processes. In other words, just because an organism seems resistant in its ability to calcify, there may be other costs or trade-offs that impact the organism negatively, and it can be challenging to find where these trade-offs can manifest, which is why a wide variety of parameters have been studied in various organisms, from metabolic rates to behavior to gene expression. And this is certainly the case of our hero of the day, the American lobster. So Rick already mentioned that lobsters have demonstrated a reaction to ocean warming. As temperatures have warmed, these animals have shifted their range northward into deeper waters. In addition, we've seen connections of warming to decreased size of sexual maturity, as well as a prevalence of shell disease and crashing fisheries in southern New England. But in terms of acidification, the benthic adults have several tools that help them kind of combat this stressor. Namely, they're able to regulate their pH around their calcifying sites. They do this. This is a figure from Kunkel et al. That shows kind of a cross section of a lobster shell, and they're able to create kind of this unstirred layer and raise the pH right against their surface. And they also have a protective waxy epicuticle that covers they're calcium carbonate structures which are already calcite, a more resistant form of carbonate compared to some of the structures that, like bivalves, for example, use a less stable form of carbonate. So although adults have these tools that help them to be better off in terms of these types of parameters than other calcifiers, we actually know a lot less about how the rapidly molting pelagic larvae might fare in future conditions. One issue is that these early stages are pelagic. They occupy the upper water column, and that region is more susceptible to rapid changes in temperature and pH than the more stable benthic environment. In addition, they're undergoing these rapid physiologically demanding molts under these changing conditions. So let me walk you through the life cycle very quickly. American lobster starts its life as an egg adhered to the abdomen of an adult female in the benthos for about 8 to 12 months. They hatch when the water reaches a critical temperature in the late spring or early summer, after which point they float up as a stage 1 larvae into the water column. And there they go through 3 larval stages, stage 1, 2, and 3. After this, they go through a large physiological, morphological, and behavioral metamorphosis into a post-larval stage 4. And this is a stage that looks a little bit more like an adult lobster that we know, but it's actually still up in the water column, moving vertically up and down, looking for suitable substrate in order to settle and eventually molt into a benthic stage 5 juvenile. Now, in optimum temperatures, this whole process takes just over a month to complete. And between these stages are important biological bottlenecks. An adult female can lay thousands of eggs, but only a few will make it through all these steps. And we just don't understand enough about how these changing water conditions might restrict these bottlenecks even further. And ultimately, it is the successful completion of all these life stages that allowed the lobster to reach harvestable size and contribute to this fishery. So this topic of larval response to acidification and warming has grown in recent years. There are 5 ocean acidification experiments on or papers on larval and post-larval lobsters. So these are pelagic stages specifically of American lobsters and their closely related congeners, European lobsters, Homarus gammarus. And these studies have come up with varying biological endpoints from a range of both end century and acute pH scenarios. And in some cases, these results are conflicting. Only 3 of these have included temperature as a joint stressor, and this is an important distinction because these 2 stressors are happening concurrently. We know that impacts of multiple environmental stressors can reveal combined effects that aren't necessarily apparent in single stressor studies, and it's becoming clear in the literature that when we ask questions about how individual species will be affected by changing ocean conditions, we really need to look at how these 2 stressors act in combination. And to make things even more complicated, these responses vary across species, life stages, and populations. So we really need to ask questions about multiple life stages and different populations. So again, there are 5 studies so far looking at OE effects on the Homarus genus. Only 3 of these have included a temperature component. Results from these studies are conflicting in some cases, ambiguous in others, but for the joint stressor studies specifically, a theme has emerged that researchers find that end century temperature has a greater effect on developmental and physiological parameters than PCO2. And this is certainly true of the work that Jess Wahle put out in 2017 and my own thesis work measuring some of these standard physiological and morphological parameters done in a lot of these studies. For example, this is a figure from a different chapter in my thesis that looked at the time it took newly hatched stage 1 larvae to reach that post-larval stage. And we had this from 2 different populations under end century predicted temperature and PCO2 levels conditions. So the blue colors represent an ambient temperature of 16 degrees Celsius. Orange represents a 3-degree predicted increase, and the lighter shades represent ambient PCO2, and as you increase our PCO2 treatments, those shading of those two colors gets darker. For this parameter, both populations showed a decreased developmental time under end-century warming, but no significant response to end-century PCO2, even though it was representing a 0.4 pH unit decrease within our treatment. So and this is pretty typical of a lot of these studies. So that led us to ask the question, are these animals just very resistant to PCO2 as a single or a joint stressor, or is the lack of strong responses to end-century PCO2s indicative of some kind of compensation on the molecular level that we have not been able to detect using the biological parameters we've measured so far? So luckily we have a tool to answer, or to look at this question anyways, and that's high throughput sequencing and transcriptomics. What this tool allows us to do is to pull RNA transcripts from our larvae raised under different conditions and look at differences in how they are up and down regulating different genes when exposed to different stressors. So if you need a little molecular bio review, DNA holds genes that code for different functions and proteins. When a cell needs a function to occur, a section of DNA that holds the code is copied into a transcript of RNA, which then exits the nucleus and is translated into a protein. This tool allows us to rapidly measure and sequence these transcripts to see which genes are being up or down regulated under experimental conditions. And if we know a little bit about what these specific genes do that these transcripts are coding for, we may be able to infer how these animals are responding and building proteins to counteract any environmental stress they may be experiencing. And by looking at these patterns of gene regulation between treatments, we may be able to detect some of those responses and trade-offs that may be less evident or even invisible in these other types of physiological studies. So our study utilized this tool to answer the question, or to determine whether end-century projected temperatures and PCO2 as concurrent stressors will affect gene regulatory response. And this is the first study to kind of look at these joint effects of these two stressors in this life stage. So how we did this is we collected egg-bearing females from Rhode Island and brought them to the Darling Center in Wahle, Maine, where we waited for a large enough hatching event from enough females. We quickly brought newly hatched eggs over to Bigelow Laboratory of Ocean Sciences in Booth Bay, where we distribute them into these tanks maintained at specific PCO2s and temperatures in a full factorial design. We used the ambient conditions of the Gulf of Maine as our control, or ambient treatment. In terms of temperature, 16 degrees Celsius is the summer average sea surface temperature in the Gulf of Maine, and those treatments are shaded blue. We use submersible heaters to bring our warmer treatments up to 19 degrees Celsius, and that represents that moderate increase of 3 degrees Celsius predicted for the Gulf of Maine. We controlled PCO2 by bubbling in air that was stripped of ambient CO2 and reintroduced at controlled concentrations using mass flow controllers. 400 parts per million represents ambient atmospheric PCO2, and hits a pH around 8.01, and 1200 parts per million in these kind of darker shaded colors is a more extreme PCO2 level that hits pHs indicative of predicted coastal pH by the end of the century, or about 7.6. You might notice that, for the purposes of this presentation, I will be referring to these conditions as ambient, temperature stress, PCO2 stress, and a joint stress when we hit that end-century temperature and coastal PCO2 at the same time. So we maintained these conditions in triplicate and monitored with daily water quality and weekly spectrophotometry and alkalinity analysis. And within these tanks, we reared our larvae up to the post-larval stage. Once this stage was reached, we allowed it 48 hours to adjust to its new body. We preserved and shipped them up to our collaborators in Canada, And there they did the RNA extractions and sent them out to be read at Genome Quebec. These reads were assembled into a transcriptome, which allows us to compare these differential expression based on treatment when there's no published genome available, which is the case for the American lobster. And for analysis, we use two different types of differential expression analysis, which I'll get to in a moment, but I'm going to start off by telling you about our results from one of these analyses known as DE-seq2. So between our treatments, this is what we found. We found that 1,108 genes were differentially expressed within these treatments relative to ambient conditions across our treatments. So this is a heat map showing a portion of our returned differential expressions. Each column represents the stressor, so this is temperature, PCO2, and joint, and the horizontal bars represent the log two-fold change of a given gene relative to the ambient treatment. So a green bar represents that transcript is upregulated, and red indicates that transcript is downregulated. So out of all the genes that we sequenced using this analysis, 18% of these genes were differentially expressed when temperature stress was present, 44% when a PCO2 stress was present, and 83% were differentially expressed when exposed to both, which is illustrated by this more vibrant column. Now, transcripts aren't that meaningful if we don't know what they do. So we ran all these transcripts through some bioinformatics software, and we found that out of those 1,100 genes, 614 could be functionally annotated. So what that means is these genes, somebody has described them in some way. We know a little bit about their function and what they do. So this is a Venn diagram just showing the proportion of annotated differentially regulated genes within each category of stressor and where the overlaps occur. Now I'm going to take this moment to remind you that joint stressor studies with larval lobster have repeatedly identified temperature as a more important driver of physiological response. But as you see here, the majority of genes were 59% of our differentially expressed genes occurred under a PCO2 stress. And almost a third, 31%, were only expressed when both stressors were present, according to this DE-seq analysis. So as I mentioned before, we did also compare our results to another expression analysis tool called EdgeR, and we did get a little bit different result. This isn't completely surprising because they use different methods of normalizing the data, and the thresholds of what they consider to be statistically significant is a little bit different. So under this, the EdgeR analysis, which are the green Venn diagrams here, for PCO2, for example, we didn't find the same weight of a PCO2 effect, but there was still a much stronger joint effect, 321 genes, than either stressor alone. So this indicates that there's agreement between these two tests that there's an interaction occurring between the stressors. Now I'm going to look at some of the genes, the annotated genes that we found by families, specifically ones that we found interesting. So the first set of genes I'm going to show you are related to immunity and antibacterial response. And I'm going to show you one stressor at a time. What we have along the x-axis is the name of these different genes, and the y-axis is the log two-fold change of the gene regulation, either in a positive or negative direction. And if there's a bar present, then that particular gene or that particular sequence was differentially up or down regulated in this treatment relative to the ambient. Yellow bars are from the DE-seq analysis, green are from EdgeR, and this represents the temperature as a single stressor. You can see not much is going on. We have a downregulation of heat shock protein over here, upregulation of the hemocyanin protein, which is involved in oxygen transport and immunity. But when we introduce PCO2, on the other hand, we see a lot more activity going on and an upregulation of much more different types of genes tied to immune function. Finally, if we introduce both stressors concurrently, we see all those genes I just mentioned light up, and in representation from both analysis tools. And what this indicates to me is that these larvae are working hard to upkeep their immune response functioning under stressful conditions. So this could be indicative of one of those compensatory responses to stress. So let's do the same thing looking at some chitin and shell formation genes. These are calcifying organisms after all. And the genes that I'm showing you were identified by Jess Wahler's thesis work on PCO2 response, specifically in post-larvae from mid-coast Maine. So in the case of our temperature treatment, we see a downregulation of several cuticle proteins and a calcification associated protein. When we introduced PCO2 as a stressor, however, we see an upregulation of chitinase and some cuticle binding molecules. So some different genes are being fired by different stressors, and when both stressors are introduced in combination, we see everything that I just mentioned, but with even more representation. Most cuticle and calcification associated proteins are downregulated. Chitinase, which breaks down chitin and helps reshape, is upregulated. And this is in agreement with some of the results from Jess Wahler's gene regulatory response to acidification stressors. What this indicates to me is that they're kind of changing the way in which they're building their shell, maybe even diverting energy away from shell building in order to put it somewhere else. These genes that I've shown you so far are obviously just a handful of differentially expressed genes that I found. We found over 600, and even more, there was about 400 that weren't even annotated, so we don't actually know their function. But what this shows us is that there's a lot of good information here, and there are a lot of things that we can use this technique for to find where some of those trade-offs might be occurring. But here are some of the key points that I took away from this study so far. Out of all the expression, more genes were differentially expressed in PCO2 stress than temperature, particularly according to that DE-seq analysis. And it seems like there's an interactive effect occurring between these stressors, resulting in much more gene activity when much more differentially expressed activity when both stressors are present. And I think that this is, there's some compensatory mechanisms at play here to keep function going under stressful conditions. And maybe this is why it's harder to detect some responses to PCO2 in these more kind of traditional morphological and physiological studies. And I think that this study provides a case for genomics work as an effective way to measure these responses to these treatments. The next step within our study is to compare these results to gene expression from larvae from a different subpopulation. So several studies have indicated that there is unique subpopulation along the range of these lobsters, and that these populations are vary in terms of thermally linked genes. If we're going to speculate how larvae might be responding to a changing climate, this ramp of diversity in thermal habitats is an important context. So this is an image showing sea surface temperature along New England coast in September. This particular study was from animals sourced from southern New England, but we also have data that is going through the bioinformatics process now from northern New England. And these regions differ considerably in terms of temperature and oceanography. This is a figure showing climatology of these two regions over the course of the year versus depth. Southern New England on the bottom here gets significantly warmer in the summer months and stratifies pretty strongly, which is relevant to these pelagic larvae, whereas northern New England maintains a cold, well-mixed water column pretty much throughout the year. So these two regions provide us kind of this this ability to use animals sourced from this region as a living laboratory in which we can ask questions about population differences in a rapidly changing environment. And with that, I'll wrap up. And thank you so much for listening. And thank you to all the folks who have funded this project. And with that, I think we're ready to take questions.
So I don't see any questions in the chat box. So if you have questions, please type them in. That was great, Maura and Rick. Wonderful presentations.
Thank you.
Thanks.
Just give you another minute to type them in. I know we have a lot of great attendees on this. That would. Okay, I see some coming in. So the first question is, when do you think it will start? Oh yeah, no, we knew that. Okay, do you believe lobster catch will increase in deeper, colder water? Richard Cliver is asking that question.
Rick, I'm going to hand that one over to you because of your deep water studies.
Yeah. Well, you can hear me okay?
Yes.
Yeah. Well, as a matter of fact, the expansion of the catch into deeper water has already happened over the past decade. We've seen a dramatic expansion into deeper water. And in fact, during the mid-2000s, we saw an expansion into eastern Maine of a big boom in larval settlement that ultimately translated into the boom in landings that really elevated the fishery to its current status. So that's definitely what we're seeing in the Gulf of Maine. And in fact, we have a paper out led by Andrew Good, also from UMaine, in global change biology that's just came out over the past few weeks. You might check that out. And another paper that's coming out in ecological applications led by another graduate student from UMaine, Noah Oppenheim. So keep an eye out for those too. But by the same token, as we've seen, an expansion into deeper and into deeper water and along the historically colder eastern coast, we've seen a recession of the catch and nursery grounds into deeper water in southern New England, and that's in part because of the inhospitable conditions in coastal areas, especially such as Narragansett Bay and Buzzards Bay and Long Island Sound.
Great, thank you, Rick. The next question is, any preliminary data for differences among populations? Diana Padilla.
We have results back from our physiological and morphological data, but I'm working through the bioinformatics right now for Down East. What I can say is that Jess Wahler's thesis work was from mid-coast versus Rhode Island, versus mine being from Rhode Island, and she saw very similar genes being differentially expressed, even though her gene expression data did not include a temperature component. But I think that there's some agreement between her study and mine in terms of some of the calcification and chitin forming genes. So that's really interesting, but it'll be especially interesting to be able to use the comparison with the joint stressors to see if there's differences there.
And I'll just jump in and add to that, Maura. So you just described some of the differences in gene expression, but there is genetic evidence from population genetics that there is differentiation along that gradient, and that comes from a series of Canadian studies over the past decade or so, first by Kenchington, who used microsatellite techniques, and then 2 papers by Beniston and co-workers from Laval University, who demonstrated differentiation at different thermally sensitive gene loci along that gradient, including Atlantic Canada, so going beyond the Gulf of Maine.
Great, thank you both. Libby Jewett would like to know, what is the next step in the research? So maybe you want to elaborate on what you provided there at the end, Maura?
Sure. So in terms of this particular project, right now we're working on getting this first piece published in terms of the results from Rhode Island animals, and then we're hoping to do kind of as we go through the gene expression results from down east animals, we're trying to also do some comparison work, kind of drawing in Jess's work from the mid-coast and those 2 endpoints from the US range to kind of compare them all. So that's a work in progress right now.
Great, thank you. So any idea how the stressor of hypoxia might fit into the mix of PCO2 and temperature? Jan Factor asked that question.
Hypoxia, yeah, that's a great question. And, you know, I don't really, I can't really speak to what kind of hypoxia stress larvae are encountering in the upper water column. It's not something that we included in this study, and we kept all of our tanks very well oxygenated, so oxygen was never a limiting factor. But yeah, when we talk about joint stressor studies, there's so many other stressors that we could include that would, you know, we'd love to include, but it makes the study that much bigger in our full factorial design. So we didn't look into that, but that's a great question.
So I missed.
Could I just jump in here quick, Esperanza, just to add a little dimension to that? You know, this question of hypoxia is really an important one as we see increasing, not only increasing temperatures, which of course is associated with lower DO levels in coastal waters, but we have the added fact that there's such a high biomass of algae, especially along coastal areas in the Gulf of Maine, that then degrade and begin decomposing, that they can become another source and compounding the low oxygen situation. And so it's definitely a concern for bottom habitat. And in fact, I know that Massachusetts, late-breaking news just reported some dead lobsters in the catch in the south part of Cape Cod Bay, and that seems to be a location where not just hypoxic but anoxic conditions have appeared this fall. Great.
Thank you, Rick. Dwight Gledhill says, really great talk, folks. I would echo that. Have you considered looking at pre-industrial CO2 levels to assess if the gene expression is tuned to current conditions or if it may already be upregulating some genes relative to past conditions?
Wow, that's an interesting question. I mean, I've never even thought about doing that. That's an interesting question. I think that kind of speaks to we're trying to kind of do this spatial gradient that kind of asks how like these different regions in terms of temperature can be predictive for a region further north that might be seeing those temperatures in the near future. But I don't know, that's a cool thought.
Okay, next question is from Beth Bisson. Great presentation, Maura and Rick. Is there any evidence of larval morbidity or mortality associated with the stress signals you are documenting here?
Yeah, so mortality is actually one of the challenges that we have with raising these, doing these larval experiments in the lab. We had a lot of, particularly within my experiments, I had a lot of problems with lobsters eating each other within the same tank. And our mortality rates were pretty high, to the point where it was hard to tell differences between treatments. I will say, Jess Waller's work looked at survival rate. We did not use that as a parameter just because our mortality rates were high enough that, you know, we were losing like 80 to 90% within our tanks. But I would look at her work for that.
Great. So the last question we have here is, are you looking at the increase in freshwater and decrease in salinity coming into the Gulf of Maine from Greenland glacial melt and its impact on the larval lobster? And that's from Richard Kleier.
I can jump in on that one, Maura. Feel free to add your own comments. But yeah, so we haven't looked at salinity as yet another stressor, and we know that you know, from some of Barney Balch's work at Bigelow that we're seeing the impact of higher precipitation levels on runoff and that lowering the salinity in the Gulf of Maine, as well as the impact from more remote impact from melting ice sheets. We don't have immediate plans to look at this, but there's some prospects out there that we're thinking about. We currently, Damian Brady and Huiji Su at the University of Maine have a new Sea Grant project funded to look at to revisit, I'll say, a biophysical larval transport model that was initially built by Huiji and Lu Inza, and they'll be incorporating the impacts of changes in the Labrador Current. I'll also say that we are also part of the Maine Arctic Network, and we'll be continuing to increase our partnership with that. And we've just been invited to be part of a NASA project that's also looking at freshening effects on circulation in the Gulf of Maine on our coastal fisheries. So several irons in the fire. I would say stay tuned.
Okay, we do have one more question. I want to be honoring people's time and the end of this, but I'm just going to go ahead and ask it. Rick talked about the decrease in groundfish being important to the huge increase in lobster. How important has the overfishing sea urchins that has led to an increase in kelp forests that are known important habitat for settled young lobster?
Yeah, so we know the depletion of groundfish has had a positive effect in relaxing predation rates. And of course the depletion of urchins has allowed for the increase in kelp. Although we don't see that kelp per se or macroalgae is necessarily great nursery for lobster. They seem to it is for cancer crabs, but we don't see lobsters recruiting to that macroalgal mat or canopy. They seem to prefer the harder substrate interstitial spaces of rocky habitat. But again, it could provide more of a habitat for larger juveniles that are on the go prior to reaching harvestable size, as sort of a temporary shelter. And of course, there are those comments about the influence of decaying algae on hypoxia, and hypoxia as being an adverse effect of that added biomass, algal biomass.
Great, thank you, Rick. So Libby Jewett just says would encourage you all to look at the data set being compiled by Dave Townsend, Damian Rue, and others who were compiling OA data across the region. Might be helpful for informing treatment levels, and you should be able to locate that through NERCUS. And that was the last question. So that concludes our webinar for today. The recording for this webinar will be posted to the NECAN website. Our next webinar will be on Wednesday, November 13th at 1 p.m., presented by Michelle Barbosa and Caroline Schweiner, I might not be pronouncing that right, of Stony Brook University, on resilience to OA and commercially important bivalves. So I want to thank you very much, Rick and Maura, for great presentations, and thanks everyone for joining us.